Hello, and welcome to Chapter 10, where we will focus in on evolution and natural selection. You've probably heard the term evolution before, but maybe you don't exactly know what it means. Well, in the process of evolution, it is going to be change in a population over time, which we will see that definition here in a minute. For this example, we are wondering if a population of flies can change their ability to resist starvation. So let's talk about it. Flies typically cannot survive past 20 hours without food. So what we've done is we've looked at this population of flies in the first generation, and we have taken the flies that survive right to that 20 hour mark and a little bit past it, and we have bred those flies together. Now we do this over and over and over, and what we start to see is that the amount of time that the fly is able to survive without food increases gradually. And again, this is generations of flies over time. So after 60 generations of doing the same process, we have now modified the population to be able to withstand starvation for 160 hours. This is an example of evolution. That is genetic change, and we'll talk about allele frequencies here in a bit, in a population over time. Now, we do want to talk about the individuals and the impact they've had on the father of evolution, which is Charles Darwin. So there's a handful of old dead guys. I do want you to know their name as well as their influence on the theory of evolution. So starting with Georges Louis Leclerc, he suggested that the Earth was a lot older than we previously believed. Now, this is kind of an important sort of understanding that I need to get across here, that at the time, the sole doctrine that explained most natural phenomena would have been the Bible. And so the Bible would have stated that the Earth was actually a lot younger than it actually is. And the fact that Georges Louis Leclerc ended up saying basically the exact opposite of the Bible is a little bit crazy, especially for the time period that he would have been saying this. Our next influence is Georges Cuvier. And what he did is he was digging around in a mine and discovered the remains of species of animals that he'd never seen before, and especially that no one had seen before in the areas that they were being found. So essentially, he was documenting fossils. So this also was kind of a crazy thing because, again, if we go back to the Bible, it would have stated that all of the living organisms on the planet at that time were put there by God, right? And that there would not have been any species outside of the species that God put there to start with. So this idea of extinction of species that used to be alive and no longer were was kind of crazy. I will show you some of these fossil records that Georges Cuvier found on the next slide. But the other individuals are Jean-Baptiste Lamarck, and he suggested that living species might change over time. Again, this kind of goes hand in hand with Georges Cuvier in that that was kind of a crazy thing to say because, again, according to the Bible, all of the living organisms on the planet at that time were put there and would not have changed from their original design. Our next individual is Charles Lyell. Charles Lyell was actually pretty good friends in life with Charles Darwin, and what he brought to the table was that geological forces would have gradually shaped the Earth and were continuing to change the Earth. So this was kind of a really cool concept, which we'll kind of come back to a little bit later near the end of the chapter, that really helped Darwin in understanding quite a bit of this idea of evolution. So here are some of those fossil remains that Georges Cuvier would have found in the 1790s. And again, imagine being in a dark mine and suddenly you come across the remains of this gigantic sloth or something kind of like an elephant. It would have been kind of crazy to see something like this. You would have thought that maybe it was the remains of some type of monster. Because again, this was not something that people had seen living in the areas that they were finding these remains. So kind of cool. Okay, you don't have to know all of this information about Darwin. Some of it's kind of just interesting information, and I will point out what I do want you to know. So Darwin was born in England in 1809 at the age of 16. He ended up going to the University of Edinburgh, and he was looking into medical studies. This was the direction that his father wanted him to go. He ended up kind of shying away from it, not really finding a lot of enjoyment in that field. So instead, he switched to study theology at the University of Cambridge. He also did not like that, so he ended up really discovering his passion, which was the study of nature. So, kind of a biological studies, if you will. So, what he did is he wanted to travel around the world and basically look at all of these natural areas, places that hadn't been like very well documented or discovered, and just see what he could see in his travels. So, he ended up becoming, and this is a little bit of extra credit for you, a gentleman's companion to the captain of the HMS Beagle in 1831. Now, the gentleman's companion is not something romantic, as it might sound. It was actually because the captain of the HMS Beagle was a little bit worried about his uncle having committed suicide years previous, and that the thought process was that maybe the suicidal tendencies was a hereditary problem and that he could inherit that way of thinking. And so Darwin was his gentleman companion or official bestie, which the captain actually really did have a strong liking for Darwin, but in a friendship capacity. Now, the area that the HMS Beagle took Darwin was kind of primarily around the equator, around the world, but the big claim to fame with Darwin is the observations he made in the Galapagos Islands. There were some patterns that Darwin observed that were kind of unexpected. He ended up looking at these physical characteristics, these traits within finches. These finches originated in the mainland, so Ecuador, that is close to where the Galapagos Islands were. Now, on the islands, there were birds, these finches, that resembled that mainland finch, but they were just slightly different. So much so to the point that Darwin ended up taking back samples of these species back to London, and he consulted an ornithologist, which is someone who studies birds, and he was questioning if they were the same species or different species, if this ornithologist could tell him anything. The ornithologist let him know that these were different species of bird, but they were on the different islands, but they all kind of resembled each other in some capacity. That was a really big observation that Darwin ended up making, leading him to believe that the primary species would have been that mainland finch, and that they would have potentially changed over time, these birds on the different islands, as the islands moved apart. So this can come into play a little bit later. We'll talk about it a little later. But that was kind of the culmination of a lot of the ideas of the individuals who inspired Darwin to look at this process that would eventually become the theory of evolution. There was another observation he made between the resemblance of fossil species and living species in the same area. For example, this, I'm not even going to try to say that word, glyptodont. Sure, we like that. The big guy on the right, which is an extinct species, which they only had fossils of, compared to the living species in the area, which was a hairy armadillo. So he was making these observations that maybe these species that are living are the descendants of these older species that are now extinct. So, one day, Darwin is reading a book, which was the Essay on the Principle of Population by Thomas Malthus, where the key point of this book was that populations would grow much faster than food could supply. This led Darwin to this phrase, and I do want you to know the phrasing because it is going to come into play later, very important. Favorable variations would tend to be preserved and unfavorable ones would be destroyed in the struggle for existence. Now, what we're describing here is natural selection. Darwin was describing natural selection with that phrase. Now, the basis of this phrase, this evolution by natural selection, would be written up into a 35-page draft in 1842. Now, Darwin was very reserved about releasing this draft of ideas to the scientific community. He did not want to be a laughingstock. He did not want to be ostracized from society. And so he kept this manuscript close to his chest and did not publish it for 14 years. That is until Alfred Russel Wallace, which was a younger biologist who was half delirious with malaria, ended up writing to Darwin after he independently identified the process of evolution by natural selection, which, if you'll remember, is the exact thing that Darwin wrote in his manuscript and sat on for 14 years. So Darwin receives this letter from Alfred Russel Wallace basically explaining that he has this idea, this natural selection by evolution idea, and he ends up kind of freaking out a little bit that, oh no, this younger biologist is going to get the credit for discovering the thing that I've already discovered. So Darwin and Alfred Russel Wallace end up getting together and they release their ideas to the scientific community, but together, which was a little bit of a safety net for Darwin because he could kind of like put it on Alfred Russel Wallace if it was not well received, except it was. So, without missing a beat, within a year, so in 1858, Alfred Russel Wallace reached out to Darwin. In 1859, just 1 year later, Darwin released The Origin of Species, which was this new dynamic view of life on Earth where we see this modification of species over time, leading to the species that we have today. So you'll notice that potentially until this lecture, you've never heard of Alfred Russel Wallace. And unfortunately, that is because Darwin did not credit him in The Origin of Species. But that's okay. Alfred Russel Wallace ended up making his own accomplishments, just not quite as big as Darwin, and never really held it against him either. Okay, so moving on to that actual process of evolution. Populations change over time, guys. From one generation to the next, we can see shifts in the allele frequencies. And the allele frequencies are kind of like a comparison. How many dominant alleles to how many recessive alleles? That is what we're looking at. So within a population, let's say of tigers, we might see that from one generation to the next, that we are seeing more white-furred tigers being born. This lets us know that for some reason it is kind of advantageous in some capacity to have white fur because we're seeing the allele frequency shifting. That is evolution. That is a population changing over time. Again, guys, individuals do not evolve. There are four mechanisms of evolution, and what happens in these mechanisms is that it is the alleles frequencies are changing, and that lets us know that evolution is taking place. These four mechanisms are mutation, genetic drift, migration, and natural selection. We're going to talk about each one individually, starting with mutation. A mutation is an alteration of the base pair sequence in the DNA of an individual's gamete-producing cells that changes an allele's frequency. So remember from a few chapters ago that DNA is made up of our A's, T's, C's, and G's. That sequence of DNA, if it is modified, any of those A's, T's, C's, or G's, then that is considered a mutation. And when we change the DNA, it changes the RNA. And if we change the RNA, that changes the protein, that physical structure. So that is a mutation, but it's also the basis of evolution in many cases. Now, the only mutations that will have an effect on the evolution of a population are going to be the cells that are reproductive. So if a mutation makes it into the DNA of a gamete, a sperm or an egg cell, and is passed to an offspring, then that is a mutation that can cause that allele frequency shift. Gentle reminder that we can't predict ahead of time which individuals will have a mutation or develop a mutation. And if that organism develops a mutation, we have no way of knowing if the mutation is going to be benign, meaning it doesn't do anything, if it is harmful, meaning it's bad, or useful, meaning it's a good mutation. Okay, on to our next mechanism, which is genetic drift. This is a random change in allele frequencies unrelated to any influence on reproductive success. So you can think of it this way. Genetic drift is this chance event that can occur that is going to affect the dominant versus the recessive alleles within a population that have nothing to do with whether or not that allele leads to a positive survival technique, if you want to think of it that way, within the population. Now, unfortunately, because genetic drift is a chance event, make sure you write that in somewhere in your notes. Genetic drift can lead to fixation, which is by chance an allele's frequency in a population reaching 100%. So the entire population has the dominant allele or the recessive allele, but that can happen. Now, the next two slides I have quite a bit of information, so I'd kind of just listen and maybe take a little bit of notes as you go. The founder effect is a type of genetic drift. The founder effect is when we have a source population that has a particular frequency of the dominant versus the recessive allele, and maybe one is more than the other. Now, within the source population, a section of that population breaks off into its own individual group to start a new population, taking with it the alleles that they have from that source population, and that then changes the allele frequency in that new population from the source population. So maybe a larger amount of the new population would have that recessive allele compared to the source population. Now, what this causes is that kind of disproportionate amount of that frequency. An example I can give you to kind of conceptualize is in Pennsylvania, there is a community of Amish. The Amish community in Pennsylvania have a higher amount of individuals that have an additional finger on both hands and potentially an extra toe on both feet. So this is called polydactylism. Words were hard for a second. So you can see in this image these individuals that have that extra finger. Now, the reason why this is an example of the founder effect is the Amish community are that newly founded population that broke off from the source population of Pennsylvania. And it just so happened that they had a few individuals in the founding population. They were the founders of the Amish community, hence the term founder effect, that had an extra finger or toe, I should say. Now, because they had a disproportionate amount of the founding population have this condition, it is now a common condition in the Amish community in Pennsylvania. So it's kind of a neat example. Okay, another type of genetic drift is bottleneck effect. Bottleneck effect is going to occur when the source population is affected and usually decimated by some type of extreme and or rapid environmental change. That is going to kill off quite a bit of the population, as well as a lot of the diversity in the genetics of that population. In fact, the example I can give you is our modern day cheetahs. So cheetahs living today, we can trace them back to a source population that would have been more genetically diverse, and as well as having a larger amount of cheetahs within the population, where now, some 10,000 years later, that population of cheetahs that we currently have is still being affected by that environmental change. So for bottleneck effect, this is going to be some type of natural disaster that causes the genetic drift to happen. So maybe a drought or a flood or an earthquake or a tornado, whatever it may be, maybe a fire. That is bottleneck effect, but it is a random chance event. So the same goes for the founder effect though too. That was completely by chance that happened. Okay, moving on to migration, which is a change in allele frequencies by individuals moving into or out of a population. This is also called gene flow, and we can see gene flow occurring not just with individuals, but their alleles. I want you to think of if you have, I don't know, siblings or cousins, or if your parents got remarried or whatever it may be, or divorced, because we are going to talk about that. If someone in your family were to have a child with someone else outside of the family, hopefully, fingers crossed, then that person leaves. Maybe they get divorced, and so that partner of your family member is no longer part of the family. Technically speaking, that person moving into your family migrated into your family, even though they left, because they had children with your family member. Their alleles are now part of your family population. So it can be individuals moving in and out of the population, but it can also be alleles moving in and out of the population. On to natural selection. This is a change in allele frequencies that occurs when individuals with one version of a heritable trait have greater success than individuals with a different version of the same trait. Now, we're going to get into this, and I'll give you an example here in a minute, but there are 3 conditions that must be met in order for natural selection to occur. The first is that the trait that we're speaking about must have variation. There must be different versions of the same trait. The other thing is that this trait must be able to be passed from parent to offspring, so it must be heritable. And then the last is that some versions of the trait need to be essentially unfavorable, and some versions of the trait need to be favorable, which is going to lead to some individuals basically being successful in surviving and reproducing, and some individuals having that unfavorable trait do not reproduce. So what we call this is differential reproductive success. To give you a little bit of imagery here, this is not my example's on the next slide, is variation of a trait, heritability of a trait, potentially bushy eyebrows, and then differential reproductive success. So something like the runt of a litter is going to potentially not live long enough to be able to reproduce. Meanwhile, its siblings that are larger and able to get to the nutrients of their mother, which the runt typically is not able to do because it cannot outcompete its siblings are going to have potentially a higher chance of reproductive success because they have a trait that is better than the runt, and that is their size. Okay, so here's that example of natural selection. So we have a variation for a trait, which is in our rabbits, the affinity for speed. So you have slow rabbits, fast rabbits, and somewhere in between a medium speed rabbit. This is a heritable trait that can be passed from parent to offspring. So fast rabbits make fast rabbits, slow rabbits make slow rabbits. And then last but not least, we're going to have differential reproductive success, which is going to result in the vast majority of the slow rabbits getting caught by predators and killed. That is an unfavorable trait. So that is going to confer poor reproductive success because, guys, you can't reproduce if you're dead. The fast rabbits are able to evade predation and survive to reproduce. So that becomes that favorable trait because it leads to a high reproductive success. And our medium speed rabbits are somewhere in the middle there. A type of natural selection is known as sexual selection. So it is not always predators that act as the selective push for evolution to occur. There can also be a push in the form of what mates desire or require within the usually male of the species. For example, female peacocks, which are kind of a brown, kind of drab color, will require male peacocks to have big, beautiful tail feathers and to really display and show them off to the girls in order to mate with them. So we can see a few examples of this with like antlers on red deer. It is a way for the males to show off and attract the females. So what this does, this push, if you will, this sexual selection, will alter how the males of a species look and potentially act. And that is completely done by the females because they won't reproduce with the males that don't meet that requirement. That means that those males will have poor reproductive success. And the males that do meet those requirements will have a higher reproductive success. So they have the favorable traits versus the unfavorable traits, and that is in order to mate. Okay, moving into the one short answer for this unit, and that is going to be a math problem in the form of the Hardy-Weinberg law. Now, I would take as many notes as you can through the next little section here, because I don't want you to get confused on the exam. So, what the Hardy-Weinberg law is an explanation that a trait will not decrease in frequency simply because it is recessive. Now remember, within a genotype, if you have a dominant allele, you will see the dominant trait. Now you might be thinking over time in a population, the dominant allele would become the only option or the only allele because you just need one to show the dominant trait. This law explains that would not happen, that we don't lose the recessive allele. So with this equation, and there are actually two equations, is P plus Q equals 1, where P is the dominant allele frequency and Q is the recessive allele frequency. So remember how we would write out P versus Q in this instance. So the dominant, oh, this is going to be so ugly, guys. Oh, that was kind of a nice arrow. The P is going to be represented as, there we go, a dominant allele. So just the allele. Q, we would represent a recessive allele as, a little b. Now remember, this is the dominant versus recessive allele frequency. Okay. Now moving into the other equation, we have p squared plus 2pq plus q squared equals 1. What we're talking about here is genotypes. Okay, and it would say genotype here if I had it typed up, but it doesn't. So P, if it is the dominant allele, I want you to remember that P squared will be the frequency of the homozygous dominant genotype. How we would write that out is 2 big B's. Sorry, that's so ugly. Q, remember, has to do with the recessive allele, where Q squared is going to be the homozygous recessive genotype, which we would write out as little b, and 2pq, which is going to be the combination of P and Q, is going to be our heterozygous genotype, which we would write out very carefully as big B, little b. So P plus Q equals 1 has to do with your alleles. P squared plus 2pq plus Q squared is going to have to do with our genotypes. Now, the only other equation that is absolutely imperative that you remember is how do we get a frequency, which is going to be that fancy little f. And we talked about this in lab. I hope you remember it. It is the, oh, goodness gracious, I should have just typed this out and I didn't because I didn't think about it, but it's fine. Here we are. It is going to be the subpart over the, there, and we got it, the total. So this is going to be one of the most important equations that you remember for the exam. Technically speaking, you don't have to inherently remember P plus Q equals 1 or P squared plus 2pq plus Q squared equals 1, but you do need to remember how to get the answer. So let's go through a problem that is in your notes. Alrighty, so what we have here is a population of kangaroo rats. There are 1,000, and let me get my little marker guy here. There are 1,000 kangaroo rats. And what's nice is these kangaroo rats have different phenotypes that result from a particular genotype. So for this guy, they are dark brown kangaroo rats, and that tells us that their genotype is homozygous dominant, and there are 16 of them. We also have the light brown kangaroo rats that are going to have the genotype that is homozygous recessive, and we have 762 of them. And then last but certainly not least, we have our heterozygous individuals, and it's nice because they have a physical representation of both of the alleles, and that is a spotted kangaroo rat. So again, they are the heterozygous, and there are 222 of them. Now, gentle reminder, when it comes to the genotypes, there are 2 alleles for each genotype. This is incredibly important, okay? So the first thing we need to do is figure out how many dominant versus recessive alleles we have within the population. All right, so that is the first thing we need to do. We are starting with P and Q, and in order to get P and Q, we have to figure this out first. So let's do it. Okay, so starting with our dominant allele, we are going to take the number of homozygous dominant individuals, which is 16, and we're going to multiply it by 2. The reason we multiply it by 2, guys, is because how many alleles do we have? 2. That gives us, for our homozygous dominant individuals, 32 dominant alleles, 32 big B's, if you want to think of it that way. When it comes to our heterozygous individuals, these guys still have 2 alleles, but only one of them, for all 222, is going to be a dominant allele, a big B. So what we do is we take 16 multiplied by 2, add 222, and that gets us 254 dominant alleles. What we do with that is we plug it into the frequency equation, because remember, frequency is the subpart. Ooh, I didn't mean to cross it out. I was trying to underline it. Subpart over the total. So we're taking 254, which is the subpart, the number of dominant alleles, and we're dividing it by 2,000, which 2,000, we got this guy because we have 1,000 kangaroo rats, and remember, each one has 2 alleles each. So 254 divided by 2,000 is 0.127. So with that in mind, let's do the recessive allele to get Q. So we're going to take 762, which is our number of homozygous recessive individuals. We're going to multiply it by 2 because each one of these guys has 2 recessive alleles. Then we are going to add 222 because each one of these guys has 1 recessive allele, so we don't have to multiply it by 2. And then what we get is 1,746. You are going to remember the frequency equation. It is the subpart divided by the total. The subpart of the recessive allele is 1,746. We're dividing that by 2,000 because remember, that total number of alleles is the number of individuals multiplied by 2 because each individual has 2 alleles each. We have 0.873 as the answer to Q. Now, we have to check our work. When we look at this equation, remember, it is going to be P plus Q equals 1. So if we add up our 2 answers for P and Q, what do we get? If you said 1, you were correct. That is how you check your work. Very important. Now, that, guys, is how you get P and Q. It is kind of a harder process because you need to know how many dominant alleles and how many recessive alleles you have in the population. Now, you might be looking at this part of the equation and thinking, ooh, this is going to suck, isn't it? The answer is no, it doesn't suck. It looks like a lot, but really, it's not. Because when you think about frequency, taking the subpart divided by the total, here we're talking about genotypes. So let's look at it. When we look at this problem, gentle reminder that P has to do with the dominant allele. That means P squared is going to be the homozygous dominant genotype. Q is going to be the recessive allele, and Q squared has to do with the homozygous recessive genotype, and 2PQ is that combination of P and Q. It's going to be the heterozygous genotype. How do we know the number of homozygous dominant versus recessive and heterozygous genotypes? It's just the number of individuals. So this number, 16 here, that is the total number of individuals that are homozygous dominant. What is the total number of genotypes? It's 1,000. So you're just going to take 16 divided by 1,000 and get 0.016, and that's the answer. That's it. Easy, right? So let's do it for Q squared. We have 762 individuals who have that homozygous recessive genotype. You just do 762 divided by 1,000, 0.762. And then last but not least, we have 222 heterozygous individuals. You take 222 divided by 1,000, 0.222. And if we add up P squared plus 2PQ and Q squared, we get 1. And that is how we check our work, how we know what the answer is, how we know we are correct. Now, guys, I'm going to give you the advice to don't round. When you do this problem, do not round anything, because what you're going to end up doing is getting a number that is either slightly over or slightly under 1. And I don't want you to do that. So just don't round. And I always ask for 3 significant figures. So a population will be at what we consider the Hardy-Weinberg equilibrium. Equilibrium just means that it is equal or balanced. You can kind of think of it that way. And it will only be at equilibrium if there is no random mating happening and that there is no evolution occurring. Now, I don't want you to worry too much about what this means and what this looks like, because we are going to have a lab that will help you understand this visually in person. Just know that Hardy-Weinberg works under the assumption that evolution doesn't occur, that P and Q will be the same from one generation to the next. So, kind of in other words, if P and Q change from generation 1 to generation 2 to generation 3, then we are quantifiably seeing evolution occur. Again, this will make more sense in lab. There are some traits that can cause individuals to have more offspring than others, and this is actually what we call fitness. So this is a measure of that reproductive output of an individual with a particular trait relative to the reproductive output of an individual of the same species with a different type of that trait. You've probably heard the term survival of the fittest, and this is kind of a misnomer phrasing, because usually we use it in the sense of competition, but this isn't really about competition. Fitness is more about how successful you are within a particular environment and how many offspring you have compared to an individual of the same species with a different physical trait. There are three important elements that go into an organism's fitness. The first is that the fitness is going to be a measure relative to other genotypes, and usually we're talking about phenotypes within a population. So again, this is going to be in the same species. The example I can give you of this element would be like our little image down here of our mice. So the phenotype that we are comparing is fur color, so a darker colored mouse to a lighter colored mouse. The next element is going to be the environment. So in the lighter environment, the lighter colored mouse is going to have a higher chance of surviving than the darker colored mouse. The darker colored mouse is going to be easily seen and probably caught by a predator. Compared to a darker environment or a habitat, the darker colored mouse is going to blend in, while the lighter colored mouse is going to stand out and probably get caught by a predator. So the environment's going to be a very important factor in this fitness. The last element of an organism's fitness is the reproductive output. So remember, you have to be alive to reproduce. So in that lighter habitat, the lighter colored mouse is going to have a higher fitness than the darker colored mouse in the same habitat because the lighter colored mouse is going to survive to have offspring. The darker colored mouse in the lighter environment is going to have a lower fitness because it has a higher chance of dying, getting caught by a predator. In the alternative environment, the darker environment, the fitness is going to change entirely and kind of reverse. The lighter colored mouse will be easily seen by predators, so its fitness is going to be lower. The darker colored mouse is going to have a higher chance of survival, so it will live to reproduce. There is a term, we have covered it in lab, and that is adaptation. Adaptation is a process by which an organism becomes better matched to survive within their environment. We saw this in lab with our mice that were the paper dots of different colors, and how well some of those colors survived in the different environments because they were able to hide better within the environment. Adaptation is going to be a feature that allows an organism to become more fit. Again, being able to survive within an environment in order to reproduce. And fitness has a lot to do with surviving within a particular habitat based on your physical trait that allows you to have more babies, more offspring. Now, as we think about this process of evolution, maybe you think that evolution would lead to an organism that would be essentially perfect. So perfectly matched to its environment, perfectly matched to evade predators, things like that. However, natural selection, while it does lead to an organism that is better suited for its environment and survival, it does not lead to the perfect organism at all. Let's talk about why. There are three factors that will prevent a population from becoming what we would classify as perfect. The first thing, and kind of one of the most important, is that environments change very quickly, whether that's from natural disasters or changes in the environment that are just naturally happening. That is an important factor that means that evolution will not be able to keep up with changing a species to match the environment because the goalpost is always moving back. The second is that variation is needed within a species for natural selection to even occur. So you can't have everything be the exact same. You have to have variation for natural selection to occur, for evolution to occur. And then last but not least, there can be multiple different alleles that lead to a trait, so they would be variations of a trait that can still have the same fitness. So you can think of our mice in our environment. A few of you had two colors that kind of tied at the end. That would be different alleles that lead to the same fitness, that same survival, and being able to reproduce. So with that in mind, you would have variation within a population that evolution could still work on. And again, if it can do that, it will still start to change that population. Again, essentially moving the goalpost back and back and back, that we would never have the perfect organism. All right, on to this term, artificial selection. Artificial selection is natural selection, but it's not natural. Artificial selection is going to be something that happens when humans breed species for particular or more exaggerated traits. This is a kind of natural process, especially when it comes to agriculture. I say natural, it's an unnatural thing, but natural for us because we want plants and animals to be what we need them to be for the purposes that we need them for. So like food or products, things like that. There are two trait categories that traits essentially can fall into. The first is a specific category where you have a trait that is one or the other. For example, white tiger fur versus orange tiger fur. There's no kind of in between. It is one or the other. The other trait category is a continuous range. An example of a continuous range would be something like height. So it's not just tall versus short, it's going to be a range in between that is different heights in between that tallest point and that shortest point. There are a handful of different types of selection, so like natural selection, artificial selection, and then these are a few more examples that I would like for you to know. This one is directional selection. This is where individuals with one extreme trait from the range of variation will have that higher fitness. And remember, fitness has to do with surviving and making babies. So in the example that I have here, this is the size of turkey breast becoming so large that that is the trait that is favorable that leads to a higher fitness, that ability to survive and reproduce. So what we see on this chart is that the larger the breast, the higher the fitness. So they have to fall in that particular exaggerated range. Now, what's kind of interesting is that this is actually an example, this specific turkey example is artificial selection as well. And that is because we have bred these turkeys to have this exaggerated feature, so much so to the point that the turkeys are not able to breed naturally. They have to be artificially inseminated in order for that trait to be passed on, but it is the favorable trait. Next is stabilizing selection. This is where individuals within a population that have an intermediate, somewhere in the middle trait are the most fit. So they have that higher chance of survival that leads to reproduction later on. The example I have for you is the weight of a newborn and how that relates to its fitness. So having an intermediate or middle birth weight, somewhere around 7 to 8 pounds, is that sweet spot. That is where we want to be. However, if you see babies that have a smaller birth weight or a larger birth weight, are more vulnerable to complications. So you don't want to see babies born at weights that are on the extreme low or the extreme high. We want it somewhere right in the middle. And then last, we have disruptive selection, where individuals with an intermediate phenotype have a lower fitness, so kind of the opposite of that last one. So this type of selection is going to prefer the extreme ends of the range. So the example I have for you is salmon, specifically male salmon size, and how they are able to sneak into nesting fields and mate. So the larger males will have a higher fitness because they are big enough to bully the other males out of the area so that they have a clutch of eggs that they can fertilize and essentially pass on their genetics. The lower end, the smaller males, are going to be small enough that they can either pass for females and the larger males don't really take notice of them, or they're small enough they can sneak in and sneak out and not get caught. So they do end up fertilizing some eggs, but in a kind of sneaky beaky type of way. The males that have that intermediate size, they are kind of in the middle range. They are not big enough to take on the larger bully fish, and they're not small enough to get in, get out, and be sneaky beaky like the smaller fish. So they have that lower fitness because it is less likely that they are going to reproduce and then pass on their genetics. Okay, we've been focusing on physical traits. However, natural selection can work on more complex traits and behavior, for example, intelligence. So in this experiment, what we've done is we've taken mice and put them into a maze and looked at how many mistakes they make and how quickly they can get through the maze. The rats or mice, or it's a rodent of some kind, that are able to make fewer mistakes are considered fast learners. They are the smart ones. And then the ones that make more mistakes are considered slow learners. These are the not as smart mice, kind of the dummies. So what we've done is we have bred the fast learners together and we bred the slow learners together. And what we see is that in each generation, you have a more exaggerated form of that trait developing. So the fast learners make even faster learners, and those faster learners make even faster learners, to the point that the rats are making very few mistakes at the end of that sixth generation. They're able to kind of really get through that maze pretty quickly. The slower learners, unfortunately, make even slower learners, to the point that at the sixth generation, we can see that the mice or rats, they end up kind of making so many mistakes, they kind of just sit down and give up in the maze, unfortunately. So we can see natural selection working on something that is more complex, like intelligence. That is something that can be passed from parent to offspring. Now, I've been telling you about the theory of evolution this whole time for about an hour here, and we want to establish, why do we support this theory of evolution? Well, there are 5 primary lines of evidence that do support the theory of evolution. We are going to go through each one individually. The first is the fossil record. So fossils are going to be a preserved species that essentially the bones have been replaced with some type of, it's not rock, it is a fossil, it is a specific thing, but it makes it to where we are able to see the preserved remains of harder structures from an organism that has died in the past. So you can kind of think of probably like dinosaurs. We have a lot of fossils from dinosaurs. Not as many as you actually think, but we do. It's kind of interesting. So I do have a little bit of extra credit here for you, and that is this fossil that was found that has a very interesting structure that has been fossilized that typically isn't, and that is feathers. I think you can probably see them pretty easily here. Now, softer tissues do not fossilize very well at all. In fact, usually it does not. So something like feathers is insanely cool to see in a fossil. In fact, this is such an important find, this Archaeopteryx. It shows a transitional form between modern birds and reptiles. And that is incredibly cool, not just because of the feathers, but also a handful of other structures like the wishbone are present. And that is showing us that link between birds and reptiles. Very, very cool. Now, just to give you an idea here, fossilization is a very unlikely event. So when it does occur, it tells us a handful of things, like where an organism would have lived. And then we can also compare that, think back to the beginning of this chapter, that within that location, we may see the descendants of that species that lived in that area. That is a very important, very cool connection, again, that Darwin was making, but that we also see as a supported line of evidence to why evolution is so well supported, especially by the fossil record. An example of a well-preserved fossil record is our modern day horse. So we can see a lot of species that have been preserved in the form of fossils from the past that led to our modern day horse. We can see a lot of transitional forms and structures in these species that would eventually lead to the structures that make up our modern day horse now. We can also look at where they were located and how they have moved from various places into others where we naturally find horses, like wild horses. Okay, another little bit of extra credit is going to be this guy, that is Tiktaalik. Tiktaalik, just like Archaeopteryx, shows us a very neat transitional feature. So Tiktaalik, just to give you a little bit of history about this animal, is that we suspect it was one of the first fish to climb out of the water onto land. So this was likely one of the first organisms that became a partially land, partially aquatic animal that would have eventually led to, over the course of millions of years, the presence of land animals. And actually, some of those land animals also went back into the water after some time. And we can see this in the sport of seeing various structures on things like whales and dolphins and hippos. Kind of neat. So this structure that is a transitional feature is actually going to be in the bones of the forelimb. So if you've ever looked at your arm, and if you can look at your arm now, if you start from your shoulder and go down to your elbow, that bone there is your humerus. If you go from your elbow to your wrist, there are two bones that make up your forearm, and that is your radius and your ulna. And we can actually see these bones labeled out on Tiktaalik, as well as the lobe-finned fishes and the early amphibians. We can see something like a humerus in that teal, and in the green, we see something like a radius and an ulna. That would be the forearm. As you move from your wrist down to your fingers, these are going to be your carpals, metacarpals, and your phalanges. We can see these represented in the bone structure of Tiktaalik in the yellow labeled bones. And then if you look at the early amphibian, that actually looks pretty similar to how we see the bones of our hand and wrist. Now, not exactly, of course, but it is very interesting that we can see this transitional feature in the bones of these animals and how they are very similar to our own bone structure now. And we'll actually talk about what that term means here in a second. But for your extra credit, I do want you to know the name of Tiktaalik and that it is that transitional organism, and it has that transitional feature of its forearm. or arm in general, front limb, because it wouldn't have been an arm at the time. Okay, on to our next line of evidence, and that is biogeography. This is the study of the distribution patterns of living organisms around the world. So this is going to be very important in understanding Darwin's finches, actually. So an example I have for you is actually the bird species, the honey creeper of the Hawaiian Islands. So we can see different species of honey creepers on the different islands. And what this tells us is that we would have had some type of original honey creeper-like species. And as the islands were closer together, they would have been able to interbreed. They would have been the same species. And as the islands start to disperse away from each other, so much so to the point that the birds would not be able to fly between the islands because they are a smaller bird. They cannot fly long distances. This leads to that diversity of these species because they become isolated. They cannot interbreed. And over time, we start to see the development of these species into their own separate different species. This is exactly what happened with Darwin's finches in the Galapagos. We start as an original species. Over time, they move away from each other, potentially because of islands moving, yes? And what we see is those birds on those islands become their own individual species over time. Okay, another example that we can see from kind of a biogeography standpoint is Australian marsupials and their placental counterparts. There's kind of a big difference here between marsupials and something called placentals. So marsupials are going to have a pouch that they hold their underdeveloped offspring in as the baby develops. So these offspring are born very underdeveloped, go into the pouch, and that is where they have milk and are able to grow over a period of time. Compared to placental animals, which are going to keep their offspring inside of their womb within a placenta, and that baby will be born fully developed, not underdeveloped. So we can see that a lot of marsupials live in Australia. This is kind of an interesting feature because they are more common there. This tells us that there needs to be that need of marsupials within that area. And then in other areas, it was more advantageous to be a placental animal. Now, this is quite neat because we can see species that are able to kind of be compared. So we've got something like a sugar glider, which is a marsupial in Australia, versus something like a flying squirrel. They're a very similar species, but they have a completely different reproductive process. We can also look at a noombat versus a giant anteater, very similar species. And then what used to exist was a Tasmanian wolf, which was a marsupial in Australia. It is no longer in existence, it is extinct, versus something like a gray wolf, which is that placental counterpart. Now, the reason why we're talking about this in biogeography terms is the marsupials are specific to Australia. So that helps support that line of evidence that evolution found, essentially, that being a marsupial was more advantageous in Australia, and in other areas it was more advantageous to be a placental animal. Now, that being said, we do have one marsupial in the United States, and that is the opossum. But there is only one. Okay, on to comparative anatomy and embryology. So this is going to lead us into this term called a homologous structure. Homologous structures, I want you to know, remember, homo means same. These are shared structures. They are the same structure across multiple species. And the reason that they are shared is because we have some type of common ancestor that would have had this trait, and this is something that we share because of that common ancestor. We inherited it from that common ancestor. And it is something that we share with other species. For example, if we look at comparing the embryological stage of a shark, a turtle, a chicken, and a human, we share two structures. That is gill pouches and a bony tail. Now, unless you're keeping a secret from me, humans don't have gills, and we also don't have a bony tail. This means that these structures are going to end up being lost as we develop. However, we have these structures during this stage of development because of a common ancestor that did have those structures. And for whatever reason, we still display that as a fetus, which is kind of interesting. Now, again, you do lose your gill pouch and your tail as you develop, but it is quite interesting that we still have this. It's kind of a leftover remnant structure. Now, kind of on to these homologous structures that we keep. So remember, those embryological stages we can look at and compare, and we share them with other organisms, but we do lose that structure. These are homologous structures. These are things like the forearm, so our humerus, radius and ulna, our metacarpals and carpals, which are our wrist, and then our phalanges. We do share similar bone structures like this with other mammals, like a horse, a bat, and a porpoise. They have a very similar bone structure to our forearm, or not forearm, I keep saying that, our arm or our front limb for a lot of these animals, or a wing or a fin for some of them as well. And that is because we have a common ancestor that had this bone structure, and it's just evolved to serve the purposes that we need them to serve. For us, it is having that arm and our fingers. For a horse, it is that forelimb and its hoof. For a bat, it's a wing, and for a porpoise, it's a fin. It's kind of interesting to see that this is something that really binds us as species. It is kind of heartwarming in a way. We are connected to each other. Homologous structures, a shared feature. Now, there are features that we have inherited from common ancestors that we no longer need. These are known as vestigial structures. They are a former but not present fitness value structure. So, for example, with these vampire bats, their diet consists of an exclusively liquid diet, which is going to be blood. However, if we look at their teeth, they have these fangs meant to kind of tear into their prey and be able to suck out the blood, but they also have something called a molar. So we have molars. Those are the teeth in the back of your mouth used for chewing and grinding. Now, for an animal that has an all liquid diet, why would they have molars? And it's because it is a leftover trait from their ancestors, because their ancestors would have had some type of diet where they had to chew. Now, they don't need their molars anymore, but it's still there. And maybe eventually they'll get rid of those molars at some point. Evolution might get rid of them, but for now, it is a vestigial trait, or a vestigial structure. We also have some vestigial structures. Your wisdom teeth are vestigial. You do not need them, and in fact, they can cause some issues, especially if you had braces growing up. The other structure that a lot of people know is the appendix. Your appendix is not a structure you need anymore. It used to house bacteria that helped us with digestion. However, we don't really need that structure anymore, and in fact, it can cause problems if it becomes infected, and a lot of people have to get their appendix removed. But it is a structure that we do not need anymore. Vestigial. So we talked about homologous structures. There is also essentially what is the opposite, which is an analogous structure, meaning that it is a structure that is not shared by a common ancestor. This is going to occur through the process of convergent evolution. Convergent evolution is going to be a type of change in a species, multiple species, that leads to the same structure, but not because they share that structure with a common ancestor. So the example I have for you would be bat wings and dragonfly wings. They both have wings, however, they do not share a common ancestor that had wings. They developed wings independently. In fact, I'd make sure that you know analogous structures mean that they are the same structure across different species that developed independently. They do not have a common ancestor that has that structure. That is an example of convergent evolution. We can also analyze things like DNA to compare the similarity between species and basically just compare that unique genetic coding. Because really, guys, a lot of our DNA, our A's, T's, C's, and G's, are things that we share with other species. We are not actually that unique as far as our DNA goes. In fact, just across humans, you share 99.9% of your DNA. That means your A's, T's, C's, and G's, the order that they go in, with the rest of the human population. There is only a small percentage that makes you uniquely you, which is kind of a weird thing to think about as far as genetics go, because we do seem quite diverse as a species. However, if we look at it on the grand scale of everything, if we all share the same common ancestors, the same thing that led to over millions of years, sometimes billions of years, that led to the species that we see now on the planet, it is, again, just like our homologous structures, something that we all share. And we can see that as proof of evolution being that answer of why do we have so many species now? And it's because we all came from that same originating source. We all share those A's, T's, C's, and G's in some order, and there's only a small amount that actually makes us uniquely human and then uniquely us. We can also look at laboratory and field experiments, but the one that I want to give you as an example, as far as something that we can see happening in a lab as well as just happening in general, is actually the example of antibiotic resistance in bacteria, and that shows a very real evolutionary progress in our lifetime, in the last like roughly 80-90 years. So let's talk about it. The first antibiotic was discovered from a mold called penicillium, which we started producing a medicine used to fight infection called penicillin. This was in 1940. Now, just take a second. I want you to guess when we saw the first lines of evidence that bacteria was developing antibiotic resistance. It was 1940. So within a year of using penicillin, we end up seeing antibiotic resistance start to develop. Now, remember that change in a population over time, from generation to generation, is what we call evolution. And that is exactly what is happening here with antibiotic resistance. So we do see that bacteria are able to, across kind of different methods, it's usually either through mutation or the transferring of genetic material from one cell to another, because they go through asexual reproduction. So they don't have any diversity really amongst a species. They do want to be genetically the same, but they can basically give a gene to another bacterial cell, sometimes to another species of bacteria. And what this can do is pass around that ability to be resistant to antibiotics. Now, what does this mean for us? It is not a good time, guys. We actually predict by the year 2050 that we will have no antibiotics that will work against any bacterial infection. And that is actually a very scary, very real concern at the moment. So there are methods in place that are looking at trying to find other ways to fight microbes. However, due to the last almost 90 years of kind of abusing antibiotics, they're too readily available. They are given to people when they are not actually sick with a bacteria. We have these broad spectrum antibiotics. People don't use their antibiotics all the way to the point that they finish that round of antibiotics. That's a very important thing to do, guys. And what that leads to is these bacteria that are not able to be killed by antibiotics. Kind of a scary thing. But again, that is that example of evolution that we can see. And with that, guys, that is the end of chapter 10. If you have any questions at all, please don't hesitate to reach out. Happy studying!